Okay, so welcome everybody to the business part of ISIB 3. We are at our first session, first scientific session, and we have our first speaker, Professor Raghavendra Gadagkar. And to introduce him, we have Professor Himinder Bharti from Punjab University, Patiala. So, Himinder, over to you.
Yeah, thank you, Sudhir. And it's my pleasure to introduce Professor Raghavendra Gadagkar, founding member and president of the Indian Society of Evolutionary Biologists. Is currently the DST Year of Science Chair Professor at the Center for Ecological Sciences, Indian Institute of Science, Bangalore. After completing his BSc Honours and MSc in Zoology from Bangalore University, he obtained a PhD in Molecular Biology from Indian Institute of Science. After this, Professor Gadagkar made what was then considered an unorthodox switch in his career and turned his attention to the study of social insects. His empirical and theoretical work on the locally available primitively used social wasp, Ropalidia marginata, being conducted for over 35 years now. He has contributed immensely to our understanding of the structure and evolution of insect societies. Gadagkar has also been exceptionally active in communicating not only his findings, but also general concepts in evolution and behavior to general audiences, especially students. So through books, that is Survival Strategies, the Social Biology of Ropalidia marginata, and popular science articles, which he's currently writing in newspaper Wire. His contributions to science have been recognized through numerous awards. To mention a few, the Cross of the Order of Merit from Germany, the Shanti Swarup Bhatnagar Award in Biology, and Schwartz Prize, to name a few. So over to Professor Raghavendra Gadagkar.
Thank you very much. Can you hear me?
Yes.
Video. Please let me know if there's any problem at any time with my audio or video. Thank you very much, Himinder, for those very kind words. Thank you, Sudhir. It's my great pleasure and honor to give the first talk of ISEB 3, which I'm very happy that we are having, and I hope in future we will have it more in person. But this time it's mixed, and I was very happy to see my friend Meva Singh brave it all the way to Patiala and be there in person. He represented all of us during the very nice inaugural session which we had this morning. Okay, so as was mentioned in the inaugural session, we are dedicating this entire conference to Edward Osborne Wilson, who unfortunately passed away recently in December 2021. Wilson clearly was perhaps the most remarkable scientist I have had the honor of personally knowing. He is known for many, many things. He lived for 92 years, and at least for 70 or 75 of those years, he has been contributing in great measure to many aspects of biology, culture, life, sociology, and many of you know about him. But I primarily like to think of him as a social insect person. He gave us 4 mammoth monographs on social insects. The Insect Societies, published in 1971, was probably the first major synthesis, modern synthesis of social insect biology. And that has been the Bible for generations of students. Even today, it is probably the most important textbook on social insects. In 1990, he collaborated with Bert Haldobler and produced another massive 735-page book called The Ants, which was again a synthesis of everything about ants. Single-handedly, much later, he published this phenomenal book, Phaidole of the New World, in which he describes 635 species of Phaidole, only of the New World. And half of those are new to science. So Wilson presents these 300 and odd new species of science in this mammoth monograph. And more recently, again with Haldobler, he's written this book called The Superorganism, The Beauty, Elegance, and Strangeness of Insect Societies. All of these are wonderful to read, and they are lasting contributions. I like to think that starting with social insects, He then moved on to many things. He famously moved on to sociobiology, in which he synthesized knowledge about social behavior in all groups of animals, particularly in the vertebrates. And this gave birth to a completely new field, which now has its own journals, its own books, its own departments. It has become a major part of the study of evolution. And Wilson actually gave birth to this discipline. But he didn't stop there. He then moved his attention to all of life and really became a spokesman for biodiversity. On the left side here, you see a picture of his book, The Diversity of Life, which tells about the richness that we have on Earth. And on the right side is a book called Half Earth, saying how we should try to save at least what is left of this. Wilson in this book makes a very bold proposal that unless we devote half of the planet Earth for preserving biodiversity, we'll probably lose this forever. But Wilson went much beyond all of this. He has written many, many books. I just will mention a few of my most favorite. He wrote a wonderful book called The Creation, in which he made a call to religious leaders. In particular, this book was an open letter to a pastor of the Southern Baptist Church, saying that religious leaders and biologists should join hands to save life on Earth. It's a remarkable book, really worth reading. For many people in this audience, I would strongly recommend another book called Letters to a Young Scientist. It really tells about how he became a scientist and how we can be inspired to be a scientist. If you want to read more about Wilson's life, there is a fiction version and a non-fiction version. Wilson actually wrote a novel called Ant Hill, which is really his life. And it's a beautifully written work of fiction. He also wrote his own autobiography, a book called The Naturalist. Of course, there are many other biographies and articles about him. So there is a lot to learn about him. I want to end this part of my initial talk by paying tribute to Wilson for his lifelong contributions, but also to Kathleen Horton. Kathleen Horton, for over 50 years, has been his research assistant, his secretary, his typist, his more recently his traveling companion. And I cannot imagine Wilson having done all of this without her constant help. Her contribution to Wilson's work is immeasurable. And I have known her also for a very long time. So I want to pay my tribute both to Ed Wilson and to Kathleen Horton. I had many times I had the pleasure of meeting him, but one thing I remember very clearly, one day I walked into the National Portrait Gallery in Washington, D.C., along with my family and with Bert Hall Dobler. And the first thing we encountered was a portrait of Edward Wilson hanging on the walls there. So Bert and I posed in front of this, and my wife has taken this picture. This is a memory that will remain for a very long time in my mind, maybe forever. I was really sad to hear that Wilson passed away in late December. I think it was the 26th of December last year. And it was very hard for me to come to terms with his loss, because this was very, very unexpected. I have tried to find ways of consoling myself. As I said in my column, which was published in the Wire Science, I tried to console myself by imagining, and here I borrowed a comment made about my favorite Urdu poet Sahib Luधियानवी. The comment is that मगर ऐसे लोग मरते कब हैं, अपने चाहने वालों के दिलों में हमेशा जिंदा रहते हैं. That's exactly how I feel about Edward Wilson. मेरी आशा है और मेरा मानना है कि विल्सन साहब हम सब के दिलों में हमेशा जिंदा रहेंगे और हमें प्रोत्साहित करते रहेंगे. उन्हीं के प्रोत्साहन से मैं अपने जीवन का सारा रिसर्च किया है और उन्हीं की प्रोत्साहन से मैं आज इस भाषण देने जा रहा हूँ. I will now switch to English and switch to my talk. As was already mentioned, I have spent most of my life working on the Indian Paper Wasp Ropalidia marginata. Today, I want to focus on a particular aspect of the social biology of Ropalidia marginata. I want to focus on queens and their successors. As all of you in the audience would know, many species of insects organize themselves into societies. which actually resemble human societies in many ways. And I would argue that they are better than human societies in some ways. They have division of labor, they have communication, they have coordination, large number of individuals live together, but they also have cooperation as well as conflict. They have two kinds of division of labor. The first one we call reproductive division of labor, where reproduction is the exclusive prerogative of one or a small number of individuals. The rest of them work as sterile workers to run the colony. In ants, bees, and wasps, the reproductive really is the queen. These are female-dominated societies. They have been called feminine monarchies, a term that an English cleric, Charles Butler, coined in the 17th century. So there is a queen, and then there are workers. Males do not really live on these colonies and partake in these societies. They just mate and die. So they're feminine monarchies with a queen and many, many female workers. Queens die and they have to be replaced, so queens have to have successors. So I want to talk about queens and their successors in this particular talk. Right in the beginning, I want to mention that many of you may be familiar with insect societies such as those of ants and those of honeybees. Ropalidia marginata is different from those insect societies in that it is what we call a primitively eusocial species, which means queens and workers are not different from each other. They are not morphologically different from each other, and they are physiologically nearly the same, which means any individual can become a queen and any individual can become a worker. And this distinction between who becomes the queen and who becomes the worker is not decided in early larval stage as it is in honeybees and ants, but actually in the adult stage. And this, of course, makes it very flexible. Queens can become workers, workers can switch and become queens, and that makes the system very, very interesting. So we call the queen and workers as two different castes. So caste differentiation or caste determination is not a matter of developmental regulation, but is a matter of social regulation. It is based on behavior of the adults, and that makes it particularly interesting. So this is the species, as I said, I and my students have worked with for a very long time. This is very convenient to work. First of all, at least in southern India, it is very common. It likes to live near human habitation, so it's very easy to find them. In fact, they live in our buildings, in our gardens. They build their nests on the windows, very easy. Secondly, they have very small nests, 20, 30, 40, 50 individuals, not tens of thousands, not millions, as in the case of ants and so on. So very easy to study. Every individual we can mark with spots of colored paint, as I'll show you in a minute, so that we can actually identify them individually. And so this is a wonderful species to study. Very early when I became interested in this, I had the opportunity to meet Edward Wilson. And he told me, this group of insects holds the key to social evolution. So that really inspired me to continue to work for my whole life. Now, because the queens and workers are not morphologically different, one of these wasps that you see here is the queen, but you will never know which one is unless you actually see her lay an egg, and the rest are workers. So that is the beauty of the primitive. But because we identify them individually and we observe their behavior for hours and hours, we of course know which one is the queen. So I will talk first about the queens and then talk about their successors. So let's begin with queens. When I began these studies, I was very intrigued by the fact that queens and workers look the same. And I wondered, are they different in behavior? So I wanted to know whether the queens behave differently from workers. How do queens behave as compared to their workers? And in order to answer this question, what I did was, since all my wasps were marked, I measured the amount of time each wasp spends in each kind of behavior. This is what we call a time activity budget. So I measured time activity budgets for all the wasps, and we then subjected this data to multivariate statistical analysis and some cluster analysis, and we were able to classify all the wasps. This is a very early paper that I published with Niranjan Joshi in 1983 in Animal Behavior. We were able to show that the wasps in a colony are of three kinds, and we call them behavioral castes, because they're not morphologically different, so they are behavioral castes. And we call them sitters, fighters, and foragers. Sitters were the wasps who spent most of the time just sitting, apparently doing nothing. Fighters were the particularly aggressive individuals showing dominance, subordinate interactions with other wasps. And foragers were the genuinely hardworking individuals who would go out and bring food. So there were three kinds of wasps, three behavioral castes. And when I did this study, I was very particular that I will not deliberately differentiate between queens and workers. If they are not different, who am I to treat them differently? So I did not worry about who is the queen and who is the worker. I constructed a time activity budget to everybody, and I did not tell my computer who is the queen, and it gave me three behavioral castes. So now I could go back post facto and ask, where is the queen? Is she a sitter? Is she a fighter? Or is she a forager? We suspected strongly that she's a fighter, because in other species of this kind which had been studied before, it was known that the queens of such primitively used social wasps rule by aggression, rule by physical intimidation. They are the most aggressive individuals, and they use physical aggression, which, by the way, we technically call dominance behavior. They use dominance behavior both to prevent workers from becoming queens, from developing their ovaries and laying eggs, as well as make sure they work. But therefore, queens must be fighters. Now, very early in my career, I was rewarded by a paradoxical result. Nothing is more valuable in science than a paradoxical result, a result that you don't expect, a result that defies textbooks. So students should not be sad that they should not think that their experiment has failed. Their experiment has succeeded because it's actually shown something new. So these queens were not fighters, they were sitters. They were lazy sitters. And so the answer to this question was that the queen of Ropalidia marginata is a non-aggressive, almost non-interactive, meek and docile sitter. And such a paradoxical result, as it always does, raised many questions. The next question we asked was, if the queen is indeed such a meek and docile sitter, how does she ever become a queen? Why do the workers respect her? If the queen is an aggressive bully, maybe they don't have a choice. But here, why should they accept her? Somebody sitting in a corner, meek, docile, non-aggressive, non-interactive. Why should I respect her and not lay eggs and work very hard? And in order to answer this question, again, early in the game, we did a different experiment. We said, if we want to know how such a meek, docile sitter queen becomes a queen in the first place, we must watch her while she's becoming a queen. And that, fortunately, is easy to do in this species, unlike in ants and honeybees, because you can remove the queen and one of the workers will become the next queen. As I already said, because there is no morphological differentiation in queens and workers, queens can switch roles and become workers, which they rarely do. Workers can switch roles and become queens, which they frequently do. And therefore, we can actually watch the process of the making of a queen. So it's very simple. We did an experiment where on the first day, we watched a healthy colony with the queen and all the workers. And here, watched means we constructed time activity budgets for all the wasps. Early next morning, we experimentally removed the queen and we observed the rest of the wasps. That is, reconstructed time activity budgets for the remaining wasps minus the queen. And on the third day, we reintroduced the queen, whom we had kept comfortably in a little glass vial with a drop of honey. She seemed very happy. We put her back at the end of the second day. She walked in and immediately began to behave like a queen. So she didn't seem to be affected by this absence of about 12 hours. And on the third day, we resampled all the wasps. We recalculated time activity budgets. And again, this experiment gave us very surprising result. There are fighters in this species. There is some dominance subordinate interaction, but by and large, I would characterize Ropalidia marginata as a fairly peaceful society. Occasionally a few bitings, lunging, chasing, but not a violently aggressive society. But as soon as we removed the queen on the morning of day 2, all hell broke loose. The fairly peaceful society became an extremely aggressive society. The frequency per hour of dominance interactions, which we measure on day 2, were, depending on the experiment, twice as day 1, 10 times as day 1, 30 to 40 times as day 1 in some cases. What was even more surprising is all of this new aggression was by one worker. One and only one worker became hyper-aggressive on day 2. Even more interesting, on day 3, when we returned the queen, as you will see here, this is the hyper-aggressive individual in the middle panel. She was showing very little aggression on day 1 in the presence of the queen. And when we returned the queen, she returned back to her original, almost original. If you wait for another day, she'll come back to her original level of aggression. This is the individual which we later discovered will become the next queen if the queen is not returned. And we called this individual a potential queen. And this is the successor that I'm going to talk about in the second part of my talk. So, as you see here, the dramatic increase in aggression by one and only one individual, and she drops that, and if you return the queen, interestingly, she will drop that aggression even if you don't return the queen in about a week's time, when by the time she becomes a new queen, because now she has to be a meek and docile, non-aggressive, non-interactive queen, and that will take about a week's time. So potential queens actually begin their careers as very aggressive individuals, and only later they become meek and docile. And that's how we can say that's why the workers respect them. But this is not a satisfactory answer. An answer to a question which is totally satisfactory is not very good, because then what do you do next? You have to go write for another grant and start a new project. Your answer should be partial, and it should tease you. It should leave something open. Now, this is nice, okay? Queens become queens because they're accepted as queens because they begin as aggressive. But it leaves open another question. How can that be true? If she's aggressive only for 1 week, how can she remain in command of the colony for the rest of her career, which may be several weeks or sometimes months, without ever being aggressive? More specifically, how does a meek and docile queen actually inhibit worker reproduction? In the other species I told you about, the queens actively go around physically harassing individuals and preventing them from reproducing, suppressing their ovaries. It doesn't happen here. So how could this queen be different? What different thing this could do in order to remain the sole egg layer, in order to retain her monopoly on reproduction without being physically aggressive? We suspected that, like in the honeybees and ants, even though this is a primitively used social species, this queen may have already evolved pheromones. Pheromones are chemical substances that animals secrete outside their body, which influence the behavior of other individuals, analogous to hormones, which we produce inside the body to influence the processes inside our body. So maybe we know that honeybees and ant queens have pheromones. Maybe she has a pheromone. And in order to do that, we did another set of experiments. And in this experiment, what we did was we brought the colony to the laboratory. We put the colony in a closed cage, again, all individuals marked. And on day 1, we studied all of them. And studied here means their behavioral profile, their time activity budgets, and the frequency per hour with which they spend with which they perform different behaviors. On the second day, instead of removing the queen, what we did was we cut the nest in half. We cut the nest half with the blade, and we put a wire mesh in between. The purpose of the wire mesh was to prevent the wasp from one side going to the other side, but to allow any volatile chemicals to go through. Having done that, we had removed, of course, all the wasps before cutting the nest. After we cut the nest and glued the nest back into the cage, we reintroduced the wasps, half of them on one side, half of them on the other side, by tossing a coin. So we pick up a wasp, toss a coin, heads you go left, tails you go right. Next wasp, heads you go left, tails you go right. Finally, we pick up the queen, do the same thing to her. She's like everybody else. We are not supposed to treat her differently. Toss a coin, heads you go left, tails you go right. In this particular depiction, she has gone to the left. So the queen and half the randomly chosen workers on the left side, the remaining workers on the right side. Now we observe both sides simultaneously, and we measure the behavioral profile of all the wasps. Now, on day 3, the equivalent of returning the queen in the previous experiment, we simply take a forceps, pick up the queen wherever she is, and move her to the opposite side, and don't touch the workers. So now the queen here was on the left side on day 2, came to the right side on day 3. So we had very clear predictions for such an experimental design. If the queen pheromone is a volatile substance, then it should go through the wire mesh, and both fragments should behave like queen colonies. There should be no difference in the behavior of wasp between day 1 and day 2. On the other hand, If the queen pheromone is a non-volatile substance, then it wouldn't go through the wire mesh. It would remain on the queen side, and the workers on the queenless side would think they have lost their queen. And they should behave like a queenless colony, which means there should be a potential queen. And if that happens, then on day 3, the potential queen on the left side should realize that their queen on the right side should realize that the queen is back, and she should now drop her aggression. But the wasp on the left side must now say, oh, we have lost our queen now, and there should be a potential queen. And that's exactly what happened. We have this particular experimental design where we put the wasp in a cage and use a wire mesh to separate fragments has been a very, very useful paradigm. And we have conducted this experiment many, many, many times. When this slide was made, this was already done 24 times. And in all 24 times, prediction 2 was upheld, which means that the queen substance is a non-volatile substance. This is what the data would look like. See, on day 1, nobody is aggressive. Neither the queen nor neither of the potential queens. On day 2, the queen is, of course, not aggressive, but on the queenless side, there is a hyper-aggressive individual. We have labeled her as PQ1 because she's the first one that became aggressive in the experiment. But on the other side, there is no aggression. Now you move PQ1 from one side to the other side, she becomes quiet, and on the other side, the new individual becomes hyper-aggressive, who was showing no aggression on the previous day, nor on the first day. Now you must realize that we can only label these animals post-factum. On day 1, we knew who the queen was, but we didn't know who was PQ1, who was PQ2. This labeling is post-factum. On day 2, we knew who the queen was, we knew who the PQ1 was, but we didn't know who the PQ2 was. That labeling is post-factum. But you see how the prediction has been upheld, suggesting that Ropalidia marginata queens appear to use non-volatile pheromones to inhibit worker reproduction. If that is true, then the question arises, again, there's a question. Every answer leads to at least 1 or 2 more questions. In fact, sometimes these questions are so serious that you don't really trust your answer. You say, let us put that answer on hold. Maybe this is what it is. Remember, I use the term here, appear to use non-volatile pheromones. We don't draw a final conclusion. We put it on hold and say, the question arising from that we need to answer, and then maybe we will have more confidence in the answer we obtained previously. So how do the workers perceive the queen? This actually is an investigation we carried on for many years, because the problem was not solved easily. Because the common sense was that workers will perceive the queen pheromone by physically interacting with her. The trouble is she is not a very interactive individual. So we tried to measure. So we tested various hypotheses. In the interest of time, I will not go through the details of the experiment. I'll just mention the hypothesis and the result. First hypothesis, workers receive the queen pheromone through direct physical interaction with the queen. We did experiments, we measured the rate, we measured how long it takes for the workers to realize, and the answer was that answer was no. So you have to put an X there. It did not work. So it is not true that they received direct interaction. Then we said maybe workers receive the queen pheromone through indirect physical interaction by a relay system. So I am in touch with the queen and you are in touch with me. I am maybe one of those favorite individuals with whom the queen rarely interacts. I receive queen pheromone and then I pass it on to somebody else. This is a relay system. Now, unfortunately, even this didn't work. Just to give you a very brief flavor how we did this, we measured the rates of interaction and we calculated the time required for the pheromone to flow from the queen to the PQ by various methods, because we know the rates of interactions. But we also know how long the PQ takes to realize, oh, we don't have a queen anymore. Time for me to become hyper-aggressive. And we match these 2 times, and the total mismatch of these 2 times. The queen recognizes the PQ recognizes the absence of the queen in 30 minutes. And these takes hundreds of minutes for information to potentially flow through physical interaction. Finally, so this didn't work. Finally, hypothesis 3, which we upheld, was the queen actually applies her pheromone to the nest by rubbing her abdomen. Okay? So that's how, and that's how now the pheromone is all over the nest, and that's how the workers can get it without waiting for this delay. And of course, the queen cannot move to the other side of the mesh, so she's only rubbing on this side, and the other side, they don't have the pheromone. So this was happened. Next question is, what is the source of the queen? In order to find the source of the pheromone, we relied on the fact that the queen is rubbing her abdomen, which means there is something on the underside of the abdomen from where the pheromone is flowing out of the body. Indeed, there is an opening there, and that connects to a gland called the diffuse gland. So we tested the hypothesis that the queen pheromone is produced and located in the diffuse gland. This is what the diffuse gland looks like. So there's a little gland here, and that opens with a duct outside, and there's a little opening here, and through that opening, the pheromone can flow out. Now, what we did when we suspected this was we dissected the wasp, we took out the diffuse gland, and we made a crude extract. And we said, if this is the pheromone, then this should mimic the queen. Instead of returning the queen and making her apply, I should be able to take the crude extract of the diffuse gland, and I should apply it to the nest, and I should quieten the PQ. The PQ should think the queen has come back. That is the bioassay we designed, and the bioassay worked beautifully. This is session 1 with the queen. So no aggression, remove the queen, heightened aggression, apply the queen pheromone, apply the crude extract of the diffuse gland, but not the queen. We just apply to the nest, and there's a significant reduction in the aggression by the potential queen. No such reduction if you apply a worker's diffuse gland crude extract, because that doesn't have the pheromone. And no reduction if you apply just the ringer solution in which you have made the crude extract, suggesting that at least one source of the queen pheromone is in fact in the diffuse gland. Having done this, then we try to see what is there in this crude extract. So we analyze the content of the crude extract of the diffuse gland using a gas chromatography machine. And we found, again, very intriguing results. Queens and worker diffuse glands contain a series of linear monomethyl and dimethyl branched alkanes with 33 carbon atoms up to that. Considering all individuals, there are at least 30 compounds which seem to be of interest. But no compound is unique to the queen or the worker. So it is a cocktail. And in many cases, this is the complication. It's a cocktail, and the wasp can tell the difference. So if you add up the areas under all of these curves, you will see that they are different. In fact, we used a computer to show that if you look at the areas of this curve, the computer can correctly distinguish a queen extract and a worker extract, making no mistake, because the composition is such. So it's a cocktail. So it's like, people who can smell different kinds of whiskey and say, this is that kind of whiskey and this kind of whiskey, because mostly it is water and mostly it is alcohol, but there are small differences. That's how the workers seem to recognize. So there is a non-volatile pheromone. So we suspect that the source of the queen pheromone is in fact in her diffuse. I want to now talk about the successors, that is the potential queens. Now, the first intriguing question about the potential queen, why did she become hyper-aggressive? Queens are meek and docile. They can manage without aggression. Why become hyper-aggressive? And why only for a week? After that, anyway, you are not. So what is the purpose of this? What is the function of the temporarily elevated levels of aggression shown by the PQ? First hypothesis. Aggression of the potential queen functions to suppress reproduction by other workers. In the first week, she doesn't have a pheromone. So she is not yet ready to become a meek and docile queen and yet be in command. So now she uses aggression, and later on she will switch to this. That's the hypothesis. Now, if that is true, if this is a temporary way of suppressing workers, then we would predict that aggression, the amount of aggression to the potential, should scale with the number of workers. More workers, more aggression is required. Or the dominance rank of the workers. High-ranking workers should receive more aggression. Or workers have different levels of ovarian development. Not enough to lay eggs, but residual amount. Now, better ovaries, more aggression. This is what we would expect if the function of this aggression is to suppress the workers. And we did not find evidence for any of this. The amount of aggression shown by the hyper-aggressive potential queen in that 1 week did not scale with the number of nestmates, did not scale with the normalized dominance rank, and did not scale with the normalized ovarian index of the workers. Now, we were kind of lost, and we needed a second hypothesis. So we put on the table an extremely unlikely hypothesis, something that I would be willing to bet on all my money on, saying this cannot be true. But we didn't have anything else. So we put it on the table. We said, aggression of the potential queen functions to boost her own ovarian development. She needs it for herself, not for others. This, of course, is very counterintuitive, but the best way to deal with the counterintuitive hypothesis is to get a counterintuitive prediction. And the counterintuitive prediction here is that a lone PQ with no nestmate should actually take longer to develop her ovaries, not less time. One would imagine that if there's no one to show aggression, all she can do is quickly put all her time and energy, start laying eggs quickly. But if she needs aggression to pump up her ovaries, then a lone PQ should take longer, and a PQ which has to spend time and energy beating up other wasps should actually do it sooner. So you can see the prediction is appropriately counterintuitive to match the counterintuitive hypothesis. Lo and behold, this is true. We actually found that when there are no nestmates, the first egg is laid sooner than if there are nestmates on whom aggression has to be shown. So the temporarily elevated aggression shown by the potential queen appears to be required for the rapid development of our own ovaries. This is quite counterintuitive, and I imagine this as influence of the physical exertion she shows in running around and beating a wasp on her physiology. Think of exercise. Exercise affects your physiology, and that's the way I'm thinking of it. And what we have done is actually we published a paper in which said we may have discovered a possible novel function of aggression. That is to boost the physiology of the aggressor. And we made the plea that people should focus on the physiology, not just of the victim, but also of the aggressor. And that will give us a complete story of how aggression works. This brings me to sort of the final question, but it took us a very long time to answer that question. And this was a common sense question. In fact, every time I gave talks, people said, as soon as you remove the queen, only one individual becomes hyper-aggressive. She is the next queen. Who is this individual? As humans, of course, we are extremely interested in knowing who is the next prime minister, who is the next vice chancellor, who is the next head of the department. We would like to know, we would like to predict. And people always wanted to know who is the next queen. And we had no idea. In fact, I always would say, it doesn't look like we can tell. It seems to be a fairly random process. We are not able to tell who the next one is in the presence of the old queen. But within minutes of removing the queen, we can unambiguously say, this is the next potential. So, question is, can we predict the successor? And the answer seems to be no. At this time, the questions became very frequent and the answers became a bit embarrassing. So I actually asked my student, Sujata Deshpande, whether she would actually take up this research. So she and Sumana and Anita, various people, they worked on this for a long time. And in fact, they showed that the potential queen is more worker-like than queen-like. And here is one example. So this is a cluster analysis with behavioral profiles. You can see that queens and potential queens are all mixed up from different colonies. Workers and potential queens are all mixed up. Only the queens are different. So they are really not different. And the story was that the PQ is not unique by any criterion, not by her dominance. Right? She's not one of the most dominant individual amongst the workers, not by any other behavior. She is not the one who has the best ovarian development amongst the workers. So we concluded that we cannot really predict the potential queen as long as the old queen is still present. And we let this be. We couldn't do anything about it. But the next student who came to my lab said, okay, we will put this question on hold. Maybe we cannot predict. Maybe it is stochastic. Maybe it is random. Maybe there is something else. But we can ask a different question. And that question, she said, was, do the wasps themselves know who the queen's successor will be? Now, this doesn't look like a very scientifically answerable question because we are saying, do they know? How can we know the mind of a wasp? How can we actually show the wasp know something or don't know something? In fact, there were a lot of people who discouraged us from pursuing this question, saying that this is not amenable to scientific answer. But this student was very determined, so she spent a lot of time trying to design experiments by which we could possibly answer this question. In fact, I remember that we had what we thought was a good experimental design, did the experiment, found the answer that the wasp actually know. But then we ourselves picked holes in our explanation and said there are alternate explanations possible for this result. So we cannot really conclude that the wasps know. And this is very important. you should not fall in love with your theory. You should not, more important, you should not fall in love with your result, which seems to explain your result. You should try and break down and knock off what you know. And we knocked off and we said, no, we need a better design. But finally, in fact, she came up with a better design. And I believe that we are able to show, scientifically answer this question. Let's make the question a little bit more precise. Wasp who would be queen? That's what I would say. That's what we're looking for. And the scientific way of framing this is it possible that there's a hair designate, even though we cannot identify it? And do the other wasps know who this hair designate is? That's the question we're asking. And in order to answer this question, we realize that what we need to do is we need to make some wasps believe that they are the next queen when they are not the next queen. And we managed to do this. Again, we use that experimental design where we bring the wasp nest to a cage, cut in half, and have a wire mesh. Now, the experiment is very tedious to do, but it is not difficult to understand. So let me explain the experiment using a simple animation. Now, this is the situation. Imagine that this is the cage. We've cut the nest in half. There's a wire mesh here, queen and half the workers on one side, queen and half the workers on the other side. Now, just reason. If there is a hair designator, by chance alone, this hair designate could have ended up. Remember, we tossed a coin and said, heads you go left, tails you go right. By this process, it is possible that the hair designate, we don't know who she is, but she is on the opposite side of the queen. It's a possibility. And if this happens, what would we expect? So here, the philosophy of the design was very different. We don't have a hypothesis to test. We say, we make an assumption. And we ask, if this assumption were true, how would the wasp behave under such a situation? So we would design an experiment and try to predict the behavior of the wasp as if our assumption is true. And if the wasp behave in that way, then we have confidence that our assumption is correct. Otherwise, we don't have. So what is the assumption we are making here? There is a hair designator. She is predetermined. And all the wasps know who she is. Only we don't know. That's the assumption. If that assumption is true, then we would expect that this hair designate should immediately become a potential queen because there is no queen and she is the hair designate. But the interesting point is, if she is the hair designate for the whole colony, she is not only acceptable to the wasp on this side, but she should also be acceptable to the wasp on the other side. So what do? We exchange the queen and the PQ, we interchange their positions, and we bring the PQ to the left side, and she should be accepted here. She should not be challenged here. We bring her back here, no problem. On both sides, she should be able to come. Nobody should challenge her. But of course, we realize that this can happen only in about half the experiments. Because by chance alone, in the other half of the experiments, the unfortunate hair designate may be sitting next to the queen. And now she can't do anything. What would you expect in such a situation? We would expect that in the other half, the best worker would become a potential. So let us call her in this experiment PQ1 because she's the first one to become aggressive in this experiment. But this PQ is not the hair designate for the colony. And the wasp on the other side will not accept her, or the hair designate on the other side because they are the hair designated. So what do? Now you interchange the PQ and Q and bring the PQ1 face to face with the hair designate, hoping that now all hell will break loose. Because now the hair designate knows I don't have a queen, I should be the next queen, but here is an imposter who is trying to be the queen. So our prediction is this should be knocked off, meaning her aggression should be knocked, and hair designate should now become aggressive. Let us call her PQ2. But that's not the end of the story. The acid test for PQ2 is now I move her to the right side. And on the right side, she should be acceptable to the wasp. Now you interchange the Q and PQ2. That's what my student did. And when she did that, she found that she was acceptable to both sides. So our predictions were PQ1, the first one to become aggressive, should be unacceptable to the opposite side about half the experiments, and a second potential queen should emerge on that side. However, the PQ2 should be accepted both sides. No PQ3 should emerge. If you cut the nest in two parts, there should never be a third contender to the third. Both these predictions were upheld. These experiments are very tedious to do, but of course, my student was very committed to this. She finally managed, after spending more than a year, maybe 2 years, 8 times. In the first 3 experiments, the first PQ held on both sides. Elder aggression was not challenged. So this is equivalent to, by chance alone, the hair designate being on the opposite side. But in the remaining 5 experiments, the first one's aggression was knocked off, a second one emerged, but she was successful on both sides. In other words, the PQ2 emerged in about half the experiments. Of course, our sample size is very small, but in about half the experiments, no PQ3 emerged. Both predictions are held, suggesting that there is a hair designate. But what is the proof that the wasps know who this hair designate is? One can guess by their behavior, but more specifically, our confidence that the wasps know who the hair designate is did not come so much from the quantity data, but from the simple qualitative result. It was remarkable that the PQ2 was never challenged. She did not receive a single act of aggression by the PQ1 or by any other workers, either when she emerged in the presence of PQ1 or when she was moved to the opposite side. And this is what gave us confidence that the hair designate was obviously known and acceptable to all the wasps, including the PQ1. So this was a wonderful period in our lab. People always used to ask me, do you know who the PQ is? And I would sort of say no. Now we could say, we don't know who the PQ is, but we know that the wasps know. Indeed, we published a paper with the title, we know that the wasps know that there are cryptic successors to the queen in Ropalidia marginata. So there is a hair designate, even though we cannot identify her in the presence of the queen, and the wasps seem to know. Now, the next student who came to the lab said, how do you know that there is a single hair designate? Is there a longer reproductive queue? PQ1, PQ2, PQ3, PQ4, are they waiting in line? Do the experiment. What this student did was, he took a healthy colony, identified the queen, removed the queen, identified the PQ, and removed the PQ, and see what happens. Now, because we can identify the PQ in 30 minutes, it's possible to do this. So when he did this, in the presence of the queen, no aggression, remove the queen, one and only one individual becomes hyper-aggressive. We call her the PQ1. Same colony, you remove her, after remaining, one and only one individual becomes hyper-aggressive, PQ2. Remove her, one and only one individual becomes hyper-aggressive, PQ3, PQ4, PQ5. So he proved in principle that. Now you may wonder, is this identification of PQ correct? Are they really going to become future queens? So my student took another set of experiments where he actually showed that these particular PQs, 1, 2, 3, 4, 5, all of them will go on to become the next queen if you don't remove them in a small set of experiments. So this is what we call a serial PQ experiment. And the remarkable thing was these PQs were never challenged. So one and only individual becomes aggressive and she is never challenged. In fact, I'll blow this up to show you that the aggression received bars, the blue bar, are essentially 0 in all the cases. And these are so high. So there is a long reproductive queue of at least 5 potential queens, but perhaps it's even longer. Maybe all individuals are. At this stage, I was quite satisfied. I said, this is wonderful. The wasps know, we don't know, and I probably become an old man, and I am quite satisfied with this. And I say, wonderful, my wasps know things that I don't know. And I was willing to be happy with this situation, saying there are always some secrets that we will never know. But young people are not like that. One of my students said, can we really not predict the potential queen? Have we tried hard enough? And this student recently finished her PhD from my lab and went to another lab, and she learned a new technique along with her mentor. This is Nitika Sharma, and this is her mentor, Noah Pinter-Wollman. And Noah Pinter-Wollman has been spearheading a method called multi-layer network analysis, where you simultaneously consider different behaviors in a network analysis. And she said, I will want to see whether I can predict the PQ based on this kind of analysis. And after a little effort, she actually succeeded. So we've just published a paper saying a reproductive hair has a central position in multi-layer social networks of paper wasps. And more recently, we have written a review article, which is in fact called On the Trail of the Potential Queen, showing how we tried and finally we have succeeded. Very briefly, there are different situations. So in the past, each social situation was examined in isolation, as we have been doing, or in any kind of network analysis. We have now used the novel analytic approach of multi-layer networks, that combines multiple social situations into a single mathematical object. A multi-layer network accounts for the differential impact of different social situations on each other, as well as on the overall global social dynamics of the colony. This network measures used to identify well-connected central individuals in traditional social networks might overlook individuals who are not important. So the traditional methods may overlook, but this one is fine. The multi-layer network has a new measure called versatility. And this reports the importance of an individual in the society while accounting for all social situations simultaneously. So for example, we use 4 situations: aggression network based on aggression, network based on prophylaxis, this is liquid food exchange, solid food exchange, and spatial overlap. Each of these doesn't tell you who the PQ is, but if you put all 4 of them together, then in fact, in the multi-layer network analysis, the number of individuals the potential queen interacted with were significantly higher than 95% of the values obtained in the randomized trials when you consider all of these simultaneously. When examining each social issue separately, there was no such strong pattern. I won't go into the details of this, but this is the graph in which you'll see this diamond, which is the real PQ, is really outside the 95% confidence rate of the distribution in all 5 colonies. But here it's all mixed up. It's not really unique. So the unique thing is really here. So we can probably identify the PQ after all. Although I must say, I will be satisfied if we can do an experiment where we collect data, apply multivariate network analysis, identify the PQ, and then remove the queen and show that our prediction is actually been held. That has not yet been done. But this is an ongoing process. Science is an ongoing process. As of now, I have a take-home message, and the take-home message is the story of queens and their successors in the Indian Paper Wasp reveals an immaculately organized society capable of adroitly managing the balance between cooperation and conflict, even in the Darwinian do-or-die situation of queen succession. I mean, nothing is more important for a worker than to become the next queen at the time that the queen has disappeared. That is probably the most important time in the life. Even in that do-or-die situation, they seem to manage with no aggression. In fact, we rarely see the wasp fight. We always were itching to see a fight. And unless we do something perverse, something unnatural, we never see the wasp fight. There's a great deal of conflict, but it is buried, it is hidden, and they seem to manage that conflict. And that is what fascinates me about this species. I want to end by thanking a very large number of students who have worked with me over the years, doing all of these. Nothing is more pleasurable than working with students. We have very little sophisticated machines and instruments in our laboratory, but we have many, many sophisticated brains, and get pleasure working with them, arguing with them, hypothesis, counter hypothesis, disagreeing with them. That's how I have been doing research all my life. And I'm very happy to have had the opportunity to talk to you about this. Thank you very much.
Thank you, Professor Gadagkar. That was really great. Shall we move to the question answers now? If you have a few minutes.
Sure.
Okay. So we'll start with the online questions first, and in the meanwhile, the offline people can give the questions to Professor Heminder Bharti, and then he will relay them to Professor Gadagkar once we are done. So Chandrakant is asking that as the PQ becomes queen, they start to become lazy seaters. Does that mean that they are investigating the total cost of energy in reproductive success by reducing their cost in fighting and foraging?
Well, first of all, we have shown, first let me give you the data. What we have shown is that if you measure PQs on a daily basis, so this is a destructive sampling, so we have many, many colonies, we remove the queen, so we look at some colonies, we study the PQ on day 0, day 1, 2, 3, 4, 5, and so on. And if you look at this, in the span of 5 to 7 days, the PQs will drop their aggression, will build up their ovaries, and will build up their pheromone profile. So their pheromone profile, which of course has to be measured in multidimensional space because it's a cocktail. So on day 0 or day 1, the pheromone profile of the PQ is statistically indistinguishable from that of a worker. On day 5 or 6, it is statistically indistinguishable from that of a queen. Now they have got this pheromone, so they don't need to show this aggression. But of course, the question is, if they need this aggression to pump up their ovaries, why do they not need it afterwards? Now, because we don't know the answer, but my guess would be that it is a kickstart. When you start your scooter, you have to kick. Once the scooter is started, you don't need to kick it anymore. So that's roughly the kind of thing I have in mind. They need to kickstart their ovaries. That's where they need this. And afterward, they probably don't need this. There may be physiological feedback which go on. Of course, I'm hand waving, as you can see. That's the best.
Right. Okay. So Puneet Deraje asked that you mentioned that the composition of the Duffer's gland secretion differs between queens and workers. How does the worker's secretion composition change when the successor worker becomes queen? Is that also a consequence of the increased temporary aggression?
We have no idea. We have three variables which are tightly correlated: change in aggression, drop in aggression, increase in ovarian development, change in pheromone profile. But we don't know which is the cause and which is the effect. We have no idea at all. These are tightly correlated three variables, but we have no idea which is the cause and which is the effect. I have no idea.
Akriti Parashar asked, can a PQ from a different nest be an heir in a nest? Like if PQ1 from nest B is placed in nest A where the queen is removed.
Interesting. So there are two things. One is that these wasps have a very strong mechanism of nestmate discrimination. They will not allow foreigners to come to the colony, and especially foreign queens or individuals which are a threat. In fact, we did one experiment a long time ago where we showed that wasps are most aggressive to foreign queens. Next aggression is towards older workers from foreign colonies, and least aggressive to the very young newborn individuals from foreign colonies. So they have a very strong sense of nestmate discrimination. They can even display this outside the context of the nest. So on the face of it, one would not expect that a potential queen from a foreign colony will come here, because just like a queen will not come, and older workers are also not accepted, and the potential queen is very unlikely. But there is a twist to this. The question is, where is the information that this is a foreign individual? So the experiment we did was the following. We take one colony, we remove the queen, find the potential queen. We take her Dufour gland. I take another colony, remove the queen, and there is now a potential queen there. We apply the Dufour gland crude extract from colony A to the queenless colony of B to see whether that PQ will respect this. She does. That's interesting. She does. So she will quite as if her own queen has come back. But if you put the real queen, she will fight with her. So we believe that the information about fertility is in one place, information about nestmateship is in another place. And in fact, we have reasonable data to show that information about nestmateship is on the body surface, on the cuticular hydrocarbons. Information about fertility is in the Dufour gland. And the real queen combines both of these. But when we apply, we only take the fertility signal and the PQ respects that. So it's a very interesting question, but the answer is nuanced.
So basically, if you take the queen and rub the body of that, then you will get a different response, is what your prediction will be. Okay. So because Sahu is asking, if we remove and reintroduce the queen multiple times, is it the only PQ1 that shows aggression multiple times? Is PQ1 fixed, or anyone can become PQ1 randomly?
So this is, I love these questions because I took out many slides for lack of time. Now I can reintroduce those ideas. Yes, this is a question I'm very interested in knowing what would happen if you do this. Again, I want to take a little longer to answer this question. People always ask me that what will happen if you replace the queen after some time? Not so much as to who will become the PQ, but what will happen. And I always said I refuse to do the experiment because I don't have a hypothesis. I don't have a prediction. Of course, what will happen is after some time, the workers will say, who are you? You are no longer my queen. Up to some time, they will accept her back. Because we know that within 24 hours, they accept her back. Maybe 48 hours, 72 hours, they'll accept her. Maybe after that, they will reject her. What do I learn from this? I didn't want to do the experiment. But later on, I had a very good reason, two good reasons to do the experiment. First is, this will test the pheromone hypothesis. Because if you wait longer, then the potential queen here will have developed her own pheromone. So now it will be a fight against equals. So it's worth doing the experiment. And the second reason is this very question of who will become the next PQ, right? With the same individual. I must say that the second reason we took first, we said okay, this experiment worth doing because it will help us to answer whether the same individual become PQ. And then we can say that the PQ is already determined and the wasp know who it is. That's the experiment we did. We got success. In fact, the same individual became PQ. But we knocked down that experiment. We said that cannot be proof because think of it like this. First time I remove the queen, let us say it's a completely random process who becomes PQ. But second time I remove the queen and put her back, that first individual who by chance became PQ first time is no longer innocent. She now has some experience and that may give her a boost to become the PQ next time, not because she's any better, but because by chance she had an experience yesterday. So that is why we discarded that experiment. That experiment doesn't prove that the PQ is predetermined. Now, because that is why we did this mesh experiment where neither PQ nor PQ1 nor PQ2 had any experience. So the same individual will become, but that for that experiment not useful. Now, in terms of this, we did a very interesting experiment. Then we used the mesh paradigm, which is a much more powerful paradigm. So we had a nest with a mesh, queen on one side, PQ on one side. Now, you let the PQ become aggressive. And then next day, you transfer the queen from the PQ, and we know that the PQ will withdraw and the queen will remain in control. We said, this is a failed experiment. We take the queen back and said, no, it's not good enough. Let's try 1 day later. 1 day later, again, PQ withdraws. This goes back, failed. We keep on doing this till the PQ says, who are you? I am the queen here. So we are waiting for a point where the two will fight with each other. And that is what I describe as a behavioral titration of the pheromone. Because now the pheromones are equal. As long as the pheromone of the old queen is superior than the potential queen, the old queen will win. If they are equal, they will fight. In fact, we are able to show that consistent with reverse parent offspring conflict hypothesis, there are 3 phases. In the first phase, the old queen wins and the PQ voluntarily withdraws. In the last phase, the old queen voluntarily withdraws and the new PQ prevails. In the middle, there is a zone of conflict where they will fight even unto death because both believe by their pheromone concentrations that they are the rightful queens. And so we actually been able to show this.
Brendan, would you like to ask something?
Sudhir, can I ask a question?
Sorry. Yeah. Can I just take Brendan's question and then I'll come to you? Hello.
Okay, great. Well, thank you. Thank you so much. This has been extremely fascinating. I'm really curious about the reproductive biology of these wasps. Are the queens monogamous? Is there worker policing of queen mating? And also just simply, how do they do it? Do they just eventually stand there and flag to attract males? So that's a set of questions. Thank you.
First, we'll take up the male part, since I've not spoken about males at all. Males close on the nest, and within about a week, they leave to lead a nomadic life, never returning to the natal nor visiting any other nest. They lead a nomadic life, pouncing on forager wasps and mate with them. So mating never happens on the nest, it happens off the nest. And it is curious because this means that the male does not know whether the female is going to mate with is a worker or a queen, is ever going to become a queen, no idea. So males try to mate quite indiscriminately with as many females as they can. Consistent with this, if you look at all the females in a colony, you will find that some random fraction of the workers are actually mated. The queen is usually mated, some random fraction of the workers are mated, others are not. Interestingly, we even found a case where the queen was not mated. And 1 or 2 days later, she unusually left the colony and later on she was mated. We know this because she started producing diploid eggs. So we have shown that not only mating takes place off the nest, but mating is a fairly random process, but mating has no influence. Mating is not necessary for an individual to develop ovaries, not necessary for her to lay eggs, viable haploid eggs, not necessary for her to become the sole egg layer in competition with other individuals who actually be mated. We've even shown that queens, PQs can become sole egg layers, start laying eggs, then go and mate later. So there is no question of worker policing because mating is sort of free. Anybody can mate. Doesn't do you any good unless you can really win in the social competition inside the nest. Now, queens we have some data that queens may mate with 2 or 3 males sometimes, and therefore the relatedness within a colony can be quite low, can be even below 0.5 in many cases. We also know from pedigree analysis that the relatedness inside a colony can be quite low, well below 0.5.
Okay. Vidya, you wanted to ask? Sorry, Professor Rangnath, would you like to ask the question? Professor Rangnath? Professor Rangnath, would you like to?
Gadagkar sir, when the queen is trying to smear the pheromone, was there any attempt by the workers to avoid it?
No, no. The queen literally sort of patrols the whole nest, walks around and rubs the abdomen. There is no interaction. She hardly interacts with the workers, and workers hardly interact with her.
How long the influence of hormone persists?
Well, it looks like it is not very long-lived, because we know that within half an hour the potential queens realize that they don't have a queen anymore. Mm. So the effect of that pheromone is very short-lived. Herein, there is a paradox, because if you look at the chemical structure of the molecules involved in that composition, they seem to be fairly stable molecules, and one would not expect them to kind of deteriorate within 30 minutes. So my hypothesis is that there is some interaction between those molecules and the external atmosphere and the nest substance, by which process it actually degrades faster than it might inside the body. So my suspicion is that inside the body it is very stable, but once it comes outside the body, interact with oxygen or whatever, and with the paper nest, then it degrades quite quickly. It's also possible, we must not forget, that the wasp may be much more sensitive in knowing what is old pheromone and what is new pheromone. Just like we smell and say, my wife will say, this upma is no good, don't eat it anymore, it's stale. I cannot make out. So the wasp may be very sensitive to know which is old pheromone, which is new pheromone. That's a possibility we should not rule out.
During the queen succession, will the process repeat?
Yes.
During queen succession, new queen comes now. Will she repeat the same thing? What the predecessor has done?
Like what? She will lay eggs, of course, and she'll be
during the queen succession, sir. New queen comes now.
Yes.
Will she repeat the processes, events, what her predecessor has done?
What her predecessor has done is she has laid eggs, which she will also do. What else you mean? Every queen will develop her ovaries and lay eggs. That's all she does. So every queen will do that. There's nothing else the queen does. She rubs her pheromone on the nest and she lays eggs. These are the only 2 things she does. And every queen will do that. She will rub her abdomen. Of course, I'll tell you, there's one more spanner in the works which we don't have an explanation for. Of course, we were interested in knowing whether the potential queen rubs her abdomen. She shouldn't, right? But she does. She does even before she has a good pheromone. We have no idea why. One of my summer interns jokingly said, sir, she's practicing, but we don't know what that means. But it is true that the potential queen rubs her abdomen even before she has the full-fledged pheromone. Now again, full-fledged is my definition. We don't know what the wasp definition is. We also don't know whether that signal of rubbing the abdomen itself could be a visual signal for the wasp. See, there are each answer leads to so many different questions. I would be happy if all the students in the audience join my lab and start trying to answer these questions.
Thank you, sir.
So, Kamla Soren has asked, what's the correlation between the multi-model network analysis prediction and the actual PQs? How were the variables selected?
That's what I said. What we have done now is we have video recordings of the nest. We know who the PQs are because later on we removed, but our video recording is before the queen was removed. And there we did the multi-rate analysis and we predicted who would be, we basically showed that the PQs stand out as different. But the definitive experiment, which we have not yet done, is to take a colony, study all the wasps, to collect the data, do the multivariate network analysis, predict who the PQ, then do the queen removal experiment and show that your prediction. This kind of experiment we have done in other contexts, but for multi-layer network, we have not yet done this. So all now, all the 5 colonies studied, the individual who happened to be the PQ could be distinguished from all other wasps multivariate network.
Okay. So Vidya has asked a question. She has said that I was wondering how long it takes for the PQ to start applying the Dufour's gland pheromone on the nest. When there is successive queen and PQ removal, do the intervals between getting successive PQs become smaller? So in some sense, the queue is not equally spaced.
Because Alok did this for 5 queens, I don't think there was much difference. There may have been a slight difference. I don't know whether it's statistically significant, but I would imagine that might happen. But first part of her question is, she seems to start rubbing her abdomen even before she has the full PQ. But this full pheromone. But this full pheromone, half pheromone, 0 pheromone is all a continuous variable. And again, being a cocktail, we really don't know what it is. But I don't know. We have not tested whether statistically significant differences were there in the time taken. I don't think we have checked that.
Okay. Ishika sorry.
The whole experiment was done in 1 day. So it was very rapid. The entire experiment was done in 1 day.
Wow. Ishika Haldar asks, can there be a chance of acoustic cues being involved?
Yes, there can be. There is evidence in some other species. In this case, not really acoustic in the sense we understand, but vibrational signals. So these insects are very sensitive to vibration. So there are, in other species, there is evidence that the queen taps the nest and that vibration spread. So in some species there are. So once we actually did an experiment to see whether that might be true, we have not so far been able to find evidence for vibrational or acoustic cues by which this information is conveyed. But we have very strong evidence that the workers recognize their queen by the pheromone. That evidence we have very clear. For acoustic, we don't have. That doesn't mean it's not possible. We just have not found a way to measure it well enough.
Vaijayanti has asked, what is the proportion of fertilized workers in a typical colony? Not sure I understand.
Very variable. We have never found 0, but we have found up to 50%, 60%, maybe 10%, 20%. As I said, so some random fraction of the worker, and that can also vary from day to day. So my impression is that when workers go out foraging, they encounter a male, and the males will mate with them. So males. Again, the male strategy is very interesting to study, and we have not studied the males enough. The males have to simply dispense their sperm to any female Ropalidia marginata with really very little hope that they will become the next. They will become the next. In fact, their sperm may be entirely wasted. So that's an angle which we have not studied, which is very interesting. So there's no pattern in the proportion of workers, and in fact, we have not also pursued that part much more, because we find that as far as we can tell, it makes no difference whether the worker is mated or not. It seems to be completely incidental.
Okay. Javed Mullah has asked, is there any interaction between the new and old queen?
There is no interaction with new and old queen, except if you do the experiment where you wait for a particular amount of time where the 2 pheromones sort of become equal, and then they fight. Before that, the new queen takes over, and the potential queen just walks off voluntarily without any interaction. Later on, the old queen walks off without any interaction, and the new queen holds. But at some critical period in between, they will actually fight unto death.
Vidya had something to ask. Vidya?
Sorry, my connection is bad. I was wondering whether the males would be able to tell who would become a potential queen.
Unlikely, because if that is true, then we would not find a random fraction of the wasps in a colony being mated. So my only inference is from that. Then,
you find a random. Okay.
And we have very often found that the potential queen is not necessarily mated, and she may go and mate later on. In fact, we did experiment in the lab where we took one mated individual and unmated individual, and both of them developed their ovaries at the same rate. It didn't make it.
Okay. Himinder, do we have any questions from the live audience?
No, but I have a question, RG. Yeah, when this transition happens between old and new queen, what kind of impact does it have on the developing stages?
this is the most amazing thing. Nothing, zero impact. No impact on the rest of the workers, no impact on the development stage. That is why I said accurately balance between cooperation and conflict. Nothing happens. Nobody is affected. The colony is not affected. That's why this is queen succession with no overt conflict. In fact, the workers don't stop working. The workers, in fact, we have evidence that in the absence of the queen, see what happens is typically when you remove the queen, the potential queen is so aggressive that she chases away all the workers. She's so aggressive that everybody flies away for a few hours. And then they slowly come back one by one and she shows aggression to them. And only by evening, all of them have come back. Now, during the rest of the day, the foragers continue to forage. We have shown that statistically, there is no difference in the rate of foraging, rate of bringing food, rate of feeding larvae, whether the queen is present or not. But there's an interesting thing. In a normal colony, when a forager comes back, she unloads the food to the wasp in the colony. And there is a method by which she knows whether more food is required. In fact, the workers use dominance behavior to tell the forager that we need more work. So if they receive the food and also are aggressive to the forager, she takes it as a signal to go and bring more. If they are not aggressive, she takes it as no need to bring more food. Now, on the day when we remove the queen, when the potential queen is crazily chasing everybody, foragers bring food, there's no one to unload. So they have two problems. One is they don't know what to do with the food they have brought. Second is they don't know whether more food is required. You know what they do? They go and feed the larvae themselves. And they go and get hunger signal information themselves and decide whether to forage or not. So there is no disruption of any kind in the loss of the queen. And this is also for natural queen turnover. That's why I said I'm impressed with the way in which, of course, there's a great deal of potential conflict, but the conflict management is really the key to the success of this society. And
Thank you.
Ajit, can I ask something? So what you are showing over here in the context of Ropalidia is that, I mean, I know that your earlier work has shown that relatedness is not a major factor and so on. And now you are essentially showing that it's essentially a bunch of chemicals and their levels on a continuous scale, etc. Now, it's very easy to imagine that something similar would probably be happening in the context of other social insects as well. So in the light of all this, where do you see the classical kin selection work? Do you think what you are doing is kind of going against it, or is it supporting that? What's your take on the whole kin selection business?
My own feeling is that in advanced societies where workers have zero reproductive options, then inclusive fitness is very important.
Mm-hmm.
We don't have anything else. But when they have both of these, you can expect this kind of dynamics. So that is, in other words, there are two ways of balancing your fitness. One is you be nepotistic. The other is you be a good politician. You try to become the next queen. So there is this interaction between indirect fitness and direct fitness. Workers have two options. One is I can play politics and become a queen myself. I can get direct fitness. The other is I can get indirect fitness. And recently we have shown that you can actually do both. For the first part of your life, you get indirect fitness and then you can get direct fitness. So when there is the interaction, so the direct fitness part, relatedness is not important.
Uh-huh.
For the indirect response. So they are playing their cards very well. They know that if I can't succeed in this, suppose I try and I fail, then my whole life's work is gone because all I've done is try to become a queen. I've not done any work. But they don't do that. They work. So they are combining indirect fitness and direct fitness in some very clever way so that their lifetime reproductive success is maximized. So I would not rule out relatedness in kin selection, but I would not give it the kind of importance that people give it, saying nothing else is important. Both are important. Both direct fitness and indirect fitness. But that only happens in these kinds of species. In honeybees, there is hardly any direct fitness. In Solenopsis, there is no direct fitness. Workers have lost their ovaries altogether. Where is the question of direct fitness? So there it becomes very important. But there's a complication there. There people say, okay, when workers lose their ovaries, what can they do? Even if there is no relatedness, there's nothing much they can do.
Mm.
So this is the stage that's critical. That is why I find it surprising that people have argued that honeybees and ants are the only species that really will tell us about social evolution. my argument would be, these are the kinds of species that will tell you about social evolution, not those who have reached the pinnacle. Those who are trying to climb and get there, those are the most interesting ones. Right? And they are in the tropics, mostly.
Right. Okay. So we will take just one last question. There are too many questions coming up as usual, but we need to end the session now. And there are, sorry?
They can email me questions.
Okay. Yeah. I mean, people, I think I will type his email ID over here. So there.
My first slide had my email ID.
Yeah, I'm sure people don't remember, but I'll give them the email ID. So there are two questions, one from Brendan, one from Puneet Deraje. Both are roughly on similar topics. So one is saying, any insights into how such a system might have evolved, especially the unique successor phenomena. And Brendan has asked similarly that, how does he imagine monogamy evolving in Ropalidia marginata? So I'm putting both questions at the same time.
By monogamy, if you mean that queen mating with just a single male, then that is largely true. Occasionally they may mate with 2 or 3 males. Mostly they mate with a single male. So, and there is only one queen per colony. So by both definitions, it is already monogamous.
Okay.
How does this system evolve? I don't know, but I would like to explore the following kind of scenario. I believe that especially in the tropics, there are lots of enemies, lots of problems, lots of parasites, lots of predators, and maintaining social harmony, especially showing social harmony to the outside world, is very, very important for success. If a colony is in trouble and the workers are fighting with each other, I think it's much easier for outsiders to gain access. And it is not just predators and parasites. Other wasps of same species are also waiting. In fact, when a colony is small, a wasp can come, invade a colony, drive away the queen, and take over this colony. So there are these usurpers. So my hypothesis is that in order to be successful in this very harsh tropical environment, not harsh in terms of climate and physical factors, but harsh in terms of social factors, maintaining harmony is very important for success. And that is why I think they have bent over backwards to put all their money on conflict management. That's what they do. And I think that's the key to survival in the tropics. This is just a hypothesis. I don't have any proof for this, but this is my hypothesis.
Okay, Professor Meva Singh has come in. Yeah. So if you will take the last question from him.
Very quick question, Raghavendra, just out of curiosity. The workers, either they get direct benefit by becoming queens or indirect benefit. But since they are not absolutely sterile, what stops them making or producing sons? I mean, are there laying workers in the colony? Are there those workers who produce sons? Of course, they don't produce daughters. Do you have the son-producing workers?
No, that is what the pheromone does. The pheromone prevents them even from developing their ovaries.
But they can still undergo meiotic cell division and lay the eggs.
No, but no vitellogenesis is possible. I think the pheromone, we don't know directly from our species, but in many species we know that the queen pheromone actually suppresses vitellogenesis and ovarian development to the worker. So actual suppression of ovarian development. You remove the queen, they will immediately develop their ovaries and produce sons. As soon as you remove the queen, in a few days they will do that. In the presence of the queen. So it is the queen pheromone which is preventing from doing that. But there is one more thing which I didn't mention. Workers have another option. They can leave the parent nest and start their own nest, either alone or with groups. So that's another avenue for direct fitness option. But if you go outside, start your own colony, it's much more risky. So again, you should see the wasps are balancing. Should I work hard for my colony and forget about direct fitness? Should I challenge the queen and try to become next queen in this colony? Or should I leave with a small group and take a risk of starting my own colony? And the possibility of success of each of these strategies, I'm sure, varies on a daily basis. Imagine a new wasp has been born who is very powerful. Then I know that my chances here are very poor, so I better leave. Or the environment has become very harsh, it becomes very dry, there is no food. Then going outside is very risky. Or the queen has changed, so now the larvae are very distantly related to me. So my indirect fitness prospects are not very good. Wasps must somehow be doing this calculus on a daily basis to decide their behavior. That's what fascinates me.
Okay.
Okay, so I guess it's 1 o'clock. I'll now request Himinder to thank the speaker, and then we can end this.
Yeah, thank you, Adi. That was a nice talk, and it's always interesting and fascinating to listen to you. Thank you so much for sparing your time.